Hi everyone, I am Dr. Anjani Devi, Associate Professor, Department of Biotechnology, Vignan's Foundation for Science, Technology and Research. As you pretty well know that I am dealing with the course Enzyme Technology, and we are moving towards Unit 5 of this particular course, Enzyme Technology. And in this Unit 5, what are the various topics which you are going to learn? We are going to learn about design of immobilized enzyme reactors. In that, specifically, we are emphasizing packed bed reactor, we are emphasizing fluidized bed reactor, and also membrane reactors. Besides, we are going to learn about bioconversion calculations in free enzyme CSTRs and immobilized enzyme reactors. Simultaneously, we are also focusing on stability, deactivation and catalytic activities of your immobilized enzymes. So, what you will be gaining after learning all these topics? You will be understanding the different immobilization methods and analyze the bioconversions in immobilized reactors. So, ultimately, you will be in position of understanding the different immobilization methods and analyze the bioconversions in immobilized reactors. Before I start Unit 5, including the enzyme reactors in which you can use immobilized enzymes, I want to give you certain important points regarding the immobilized enzymes. As we are talking about enzymes, as we pretty well know that enzymes are biological catalysts, they are proteinaceous in nature. And these enzymes are important because they will be increasing the rate of reactions without disturbing the equilibrium of the reaction. In the previous units, in Unit 2, we have discussed about various sources of enzymes, how you are going to extract the enzyme, clarify the enzyme and purify the enzyme. And in Unit 3, we have studied about enzyme kinetics. And there we have talked about free enzymes. Why we need enzyme? We need enzymes, we require enzymes in order to carry out all the metabolic activities that are taking place in our body. And these enzymes are having specific characteristics. They are stability of the enzymes, active site of the enzyme, and these enzymes are stable under the optimum conditions such as optimum pH, optimum temperature. This enzyme activity depends upon the concentration of your substrate, concentration of your enzyme, etc. If these environmental conditions are under the optimum conditions of this particular enzyme, they will be stable. Otherwise, your enzyme is going to get unstabilized. If at all your enzyme is denatured, you can't further carry out the reaction. You can't convert the substrate into product. So, enzyme stability is very much important. And in order to improve the stability of the enzymes, in order to enhance the half-life of the enzymes which are used in the reactor where drastic conditions such as high temperature, high pH are maintained, we are going for immobilization of enzymes. What is immobilization? Immobilization of these enzymes is nothing but restricting the movement of the enzyme to the specific area. Because of this protection of this enzyme, we can use the enzyme for number of batches of reactions. That means you can make your process economical. Simultaneously, in order to immobilize the enzyme, we discussed about various physical and chemical methods. In the physical methods, we have discussed about adsorption method. We have discussed about entrapment method. Under chemical immobilization techniques, we have discussed about cross-linking, covalent binding, etc. So, with this background information, we are now going to study about various reactors where you can use these immobilized enzymes. So, enzyme reactor. What is an enzyme reactor? How it looks like? The enzyme reactor will be like a vessel or it may be a single vessel or it may be constituting a series of vessels used to perform a desired conversion by enzymatic means. So, enzymatic reactors are the vessel or series of vessels used to perform a desired conversion of your substrate into product by the action of your enzyme. There are several important factors that determine the choice of the reactors for particular process. You can't use all the reactors for all the process. You have to choose which sort of reactor is suitable for your process. So there are various parameters, various important factors that determine the choice of your reactor according to the particular process. So cost of a predetermined productivity within the product's specifications. This includes cost associated with the substrate, downstream processing, labor, as well as overheads and process development. So, whenever you are choosing a particular reactor for a particular process, you have to bear in mind these factors. First important thing is you have to think about the cost of the particular substrate which is associated with the process. Moreover, what are the different downstream processing you are using in your process in order to convert your substrate into product and purify your product? So you have to bear in mind about the number of downstream processes. We have to bear in mind about the downstream processing. Moreover, we have to think whether that particular process is labor intensive process or not. And we have to think about these overheads and process developments. So we have to think about majorly these factors whenever you are planning for a reactor for a particular process. And then we have to discuss about design and configuration of immobilized enzyme reactors. How we are going to design an immobilized reactor. How we are going to configure a enzyme reactor suitable for your immobilized enzymes. Irrespective of the physical forms of the immobilized enzymes such as solid phase reactor or membrane reactor or solid phase film reactor, we are considering three important factors of immobilization procedures such as we have to think about what are the various properties of the enzymes which you are using in the process. What are the type of support you are using for immobilizing this enzyme? And what are the method of the support activation and enzyme attachment? So you have to think about these three important features before you start immobilizing the enzyme. First you have to think what is your enzyme? What is the nature of your enzyme? After knowing about the properties of your enzyme of interest, you have to think what is the support or what is a carrier which is compatible for this enzyme and how I am going to activate my support, how I am going to link up my support with the enzyme. So I have to think about all these three important factors before we start immobilizing an enzyme. So as I've told you that knowing the properties of enzyme is a very important step. So what we have to know about, what is the important information which you need to know about the enzyme which you are going to use. You need to know about the classification of your enzyme. You need to know about the systematic name as well as the EC code of your enzyme of interest. Then you need to know about what is the type of reaction that is catalyzed by the enzyme. Then you need to know what are the various sources from which you can obtain your enzyme of interest. What are the various sources means you have to know what is a plant source, whether you are getting your enzyme from the plant source, animal source or microbial source. And moreover, you have to also think about the amount of enzyme you are getting from each source. And then you have to decide according to the abundant availability of your enzyme from a particular source, you have to confirm that source. Moreover, you have to think about whether the enzyme is extracellular or intracellular. Then you have to think about whether the process is economical or not. The process of enzyme extraction and purification is economical or not, then you have to, by bearing in mind all these characteristics, you have to choose a particular source. And then you have to know about the physical forms of your enzyme or the structure of your enzyme. What you need to know about the information about the active site of enzyme, catalytic nature of the enzyme. So you need to know the nature of the enzyme which you are going to use. Moreover, you need to know the purity, how much purity you require. For example, if you are going for the production of high value, low volume product, that means you are going to get a product in less quantity, but the value of the product is high. If at all you are going for production of high value, low volume product, then you have to take care of purity. Your enzyme should be ultra pure. That means you have to use very sensitive techniques for the purification. So, you have to decide the requirement for your enzyme, how much purified enzyme you require. According to that, you can choose the number of downstream processing steps. And you have to also confirm what are the various purification steps that you are going to use for the purifying your enzyme. You need to know the information about the catalytic activity of your enzyme. Moreover, other details like what is the optimum range of your enzymes, optimum pH, optimum temperature, and the stability of the enzyme, half-life of your enzyme of interest. After knowing this information, you have to then step into the next phase, that is nothing but your selection of your support. Once you know the characteristics of your enzyme, then only you have to go for a selection of proper support in order to immobilize your enzyme of interest. Besides those properties, you should also have the information of enzyme activity. As I have told you that we need to check or we need to select that particular source from which you are getting maximum amount of your enzyme. As you can't directly measure the enzyme concentration, you can measure enzyme activity. You have to check the activity of the enzyme which is obtained from the various sources, and then you have to select that particular source from which you are getting the enzyme with highest activity. And moreover, I have told you that the enzyme activity and the specific activity will be depending upon the number of purification steps you are using and the sensitiveness of your purification method. And then this information is very much essential because it allows us to compare the enzymes from various sources. Moreover, from the analytical point of view, catalytic activity of the enzyme is very important because it has direct relation on the sensitivity. Moreover, the knowledge of units of enzyme activity and the specific activity is also very important. So, details of your enzyme support. Enzyme support can have a critical effect on the stability of the enzymes used and the efficiency of the enzyme immobilization method. When we are discussing our unit 4, there we have discussed that the immobilization method should be in such a way that it should not hamper or it should not reduce the stability of the enzyme. The bondings that are involved in binding your support and your enzyme, that bond should not be between the active site of the enzyme as well as the support. The bond should be with any other reactive group except your active site. That means the linkages should not be present between your support as well as the active site of the enzyme. If your enzyme is bound to the support through a bond which is linking your active site with the carrier, then there is every chance that the activity of your enzyme can get reduced. So, in order to properly immobilize the enzyme, you have to choose the support as well as the method through which you are immobilizing your enzyme to your support. So, the activity of the enzyme depends upon the immobilization method. used for the enzyme immobilization. Even though it is very difficult to predict in advance the suitability of the support for a particular enzyme. So, criteria for selection of the support are, first one, the support should be soluble in nature. The support material which you are taking should have solubility. It should be soluble in water. Moreover, it should have high binding capacity to the enzyme. It should bind to the enzyme very strongly. Not that much strongly that it can hamper the stability, but it should have strong bonding so that carrier won't be getting separated from the enzyme during the reaction process. So, there should be a good binding capacity between your enzyme and your support. Moreover, it should be chemically inert and mechanically stable. Your support should be inert. It should not be in such a way that it will be reacting with your enzyme. So, if you are selecting your support which is inert, then the support won't be reacting with your enzyme molecule. That's why you need to select your support which is inert and it is mechanically stable. And people need to know that there is no universal support that fulfills all the requirements. There are number of supports. Every support will be having its own advantage and disadvantages. So you need to go for trial and error method. You need to check your enzyme immobilization with various supports. Then you need to measure the enzyme activity. You need to select that support where your enzyme activity is not getting reduced. Your enzyme is stable. You need to select that immobilization system which is allowing your enzyme to stay in a stabilized condition. So, you need to know that there is no universal support for enzymes which will be fulfilling all the requirements. Final choice of the suitable support is a compromise for each particular enzyme and experimental system. As I've told you that you need to go for different trial and error methods, then you can finalize one particular support suitable for your enzyme. There are three different types of support materials such as the first type is your hydrophilic biopolymers which are made up of natural polymers such as cellulose, dextrans and agarose. Coming to the second category, they are nothing but lipophilic synthetic organ polymers such as polystyrene, polyacrylamide and nylon. Coming to the third type of your support is inorganic materials. Example, iron oxide. In each case, the chemical structure of the support must be defined along with the quantitative, physical and chemical data relevant to the above factors. So, as I told you that you need to check the compatibility of your support with your enzyme. So, after knowing the properties of your enzyme, which is the first step, you are going towards the selection of the support, and then as a third step, you are going for activation of your support. So, support activation and enzyme attachment. So, you have taken a support, you have selected a support, you are ready with your enzyme. Once you are ready, before immobilizing your enzyme, in order to immobilize your enzyme onto the support, you need to activate your support. Sometimes there is every need that you have to incorporate certain functional groups onto your carrier which has capacity to bind with the enzyme. So, that process is called as activation of your carrier. So, the 5 important immobilization methods are adsorption. We have studied about covalent binding. We have studied about entrapment, cross-linking and membrane confinement. That's nothing but is a part and participle of encapsulation, entrapment. So, we have studied about all these immobilization methods. Among all these methods, mostly used method is nothing but your covalent binding method. So, covalent binding method is most widely used in order to immobilize your enzyme with your support, or immobilize your enzyme onto your support. In continuation, an active material has enzyme reactive functional groups that is covalently linked to the inert surface of the carrier. Sometimes during activation of the material, there is a probability to add certain functional groups that can bind to the enzyme. The most important factors that have been taken into consideration are stability of the resulting bond between the enzyme and the support, the local environment of the enzyme, and the potential loss of activity of the enzyme. So, while you are immobilizing your enzyme, you have to bear in mind about the three important factors. The first one is your enzyme should be stable. Your immobilization process should not hamper the stability of the enzyme. It should not change the local environment of the enzyme. It should not reduce the activity of the enzyme. So, in order to summarize, the important features for immobilization are: you need to know the chemical nature of your enzyme as well as your support. Then you need to know the chemical nature of the coupling reagents that are incorporated during activation of your support in order to bind to your enzyme. And what are the various experimental conditions where you are going to use your immobilized enzyme. Besides, you need to know that sometimes these activation methods, immobilization methods, they will be destabilizing the enzymes. So you need to check and use that method, that immobilization method, where your enzyme is not going to get deactivated. Besides, the stability of the enzyme with respect to the time, temperature, ionic strength, impurities, pH, and the storage stability are also very important factors. You need to study about these factors also before you start immobilization of your enzymes and using those immobilized enzymes in the reactor. I want you people to work on these questions so that you can assess how far you have understood from today's session. I followed these links for today's session. So I want you people also to follow the links in order to gain more information about the topics covered in today's session. Thank you.